Hi everyone. So today we are going to discuss polymerase chain reaction or PCR. Now, polymerase chain reaction is extremely important when it comes to different kinds of biotechnological procedures. So for example, if I want to clone a particular gene, then for gene cloning, you will be isolating the gene from the DNA, let's say human DNA. However, the amount of the gene that you isolate might not be enough to conduct your experiments, right? So in that case, we need to amplify the amount of DNA that you have. PCR comes very handy when we want amplification of DNA. Similarly, it can be used in genetic testing. It is very widely used in disease detection. For example, recently when the pandemic was happening, COVID was detected using the PCR technique. So let's understand what PCR is. If you remember what replication is, what happens in replication is that in your body, the DNA gets separated, the two strands form a replication fork. So like this. The two strands will form a replication fork, and at the replication fork you will have formation of DNA. Correct? Yeah, so that's what replication is, and for replication to happen, we know that a particular few enzymes are very important. When it is happening within the cell, obviously you need some thing to separate these two strands, because if you remember, there is an ATCG complementarity, right? Hydrogen bond formation, which keeps both the strands of DNA together. In our system, there are enzymes like helicases, right, and topoisomerases, which help in unwinding the DNA, separating the two strands, and also keeping it from getting supercoiled behind, right? And then enzymes like DNA polymerase will come and make a new DNA, starting with a primer always. DNA polymerase cannot act from scratch, so you need a primer which the body provides. It's an all 10 to 15 base pair sequence which will sit, and then it will happen, right? One more interesting thing that happens is that the new DNA strand that is produced by the polymerase always is a 5' to 3' strand. Therefore, you get one strand, which is the original 3 to 5 reading, will give you the continuous strand, whereas the opposite one will give you a discontinuous bits of fragments known as the Okazaki fragments. This concept, when done in a laboratory in a machine, it's known as PCR or polymerase chain reaction. Now, in polymerase chain reaction, however, we do not use enzymes like helicases and ligases. We use the concept of temperature. Temperature denatures the hydrogen bonds, and therefore temperature can be used to cause separation of the strands. So then again there are three steps. One is denaturation, elongation and annealing. Denaturation usually happens at a temperature which is known as the Tm, which is directly proportional to the amount of CG bonds, because CG is the triple bond. So basically one DNA strand which has a higher amount of CG bonds or CG base pairing will require a higher temperature, whereas with lesser will require lower temperature. On an average, it's about 94 degrees Celsius. For 0.5 to 2 minutes, we would expose the DNA strand, and that would lead to the formation, not really of the replication fork, because replication fork is formed in the case of a large segment of the whole DNA. But here, because we take smaller segments, there is going to be, so if this was the original, then there is going to be strand separation. They will become separate. Now next, what we need are primers. Primers are artificially supplied in this case. So you will obviously know the sequence of your DNA segment, the initial 10 base pair sequence. So you will make a primer, oligonucleotide primer, which you will supply in your medium. Next comes polymerase. The biggest problem with this was the polymerase, because most of our bodily enzymes get denatured beyond an optimal temperature of 35, at best at 40-45. But in this case, we needed the enzyme to be working functional at least a 72 to 80 degrees Celsius temperature. We found a particular kind of bacteria known as the Thermus aquaticus. It grows in the hot water geysers of the world. When we isolated the DNA polymerase of this particular bacteria, we found that this enzyme can withstand very, very high temperatures. Obviously, the bacteria lives in hot water and it obviously replicates because that's how it reproduces. So, when that polymerase was isolated, it was named the Taq polymerase. And Taq polymerase is used for PCR reactions. So then the medium will also have Taq polymerase. So what happens is that the primer will sit, Taq polymerase will start to work. So now you supply your medium with DNTPs, right? So basically DATP, DGTP, DCTP, DTTPs. And just like a normal polymerase would work, it would keep adding them to your DNA segment. Once the elongation has happened, so the whole DNA segment has been copied, you need to anneal here, you need to join here, right? And you need to let the hydrogen bonds form again so that you have a total strand. For annealing, we use a temperature of about 54 to 60 degrees Celsius, where the DNA strand re-anneals, and now you have from 1, you have 2 DNA strands. So in one cycle, what happens from 1, you get a total of 2. It doubles, correct? So from 2, you will get a total of 4. And therefore, if you do this cycle for n number of times, you will get the doubled amount of DNA. This helps in DNA amplification. It helps in making up DNA libraries, so we can work on those later. And thus, PCR is a very, very important step in biotech. I hope this much was clear. I'll see you in the next video. Thank you.